For decades, scientists thought they understood how plants fed themselves. Roots absorb minerals from the soil, leaves capture sunlight. Simple, clean, done. They were wrong. Beneath every garden, every farm, every wild forest floor, something far stranger is happening. Your plants aren't just absorbing nutrients passively, they are actively hunting microbes, pulling living bacteria into their root cells, stripping them of minerals, and then, and this is the part that changes everything, ejecting what's left back into the soil to start the cycle again. This process has a name. Scientists call it rhizophagy, and once you understand it, you will never look at bare soil, synthetic fertilizer, or a bag of sterile potting mixed the same way again. The word rhizophagy comes from the Greek, rhizo for root, phagy for eating, root eating. But what's being eaten isn't the root itself, it's the microbial life the root deliberately captures. The foundational research on this mechanism was led by plant biologist Dr. James F. White and his colleagues at Rutgers University. Their work, published in peer-reviewed plant science literature over the past decade, documented something that turned conventional plant nutrition theory on its head. Plants aren't passive recipients of whatever minerals happen to dissolve in soil water. They are active participants in a sophisticated underground economy, one where they recruit microbes, exploit them, and recycle them in a continuous loop. Here's how it works at the cellular level. Plants release chemical signals from their root tips, specifically reactive oxygen species, including superoxide, that strip the protective cell walls from bacteria living in the soil. Once stripped, these microbes become small enough to be absorbed directly into the root hair cells. Inside those cells, the bacteria are transported inward toward the shoot tissue. As they travel, the plant extracts minerals from them. Nitrogen, phosphorus, potassium, zinc, copper, the full spectrum of what a plant needs to grow. Then, remarkably, the plant ejects the surviving microbes back out through the stomata, the tiny pores on the leaf surface. Those microbes fall back to the soil, rebuild their cell walls, scavenge more minerals from the surrounding environment, and eventually find their way back to the roots, where the cycle begins again. Think about what that means. The plant isn't waiting for rain to dissolve minerals and carry them passively to the root surface. It's sending out a workforce, cycling that workforce through its own body, and returning them to the field to gather more. This isn't passive chemistry. This is active biological farming happening at a scale we can barely see. And here's what makes this genuinely unsettling if you've been gardening for years. Most of the practices we consider good, sterilized growing media, high-dose synthetic fertilizers, frequent fungicide applications, actively disrupt this cycle. Not because they're evil, because nobody told us it existed. Drop a 1 in the comments right now if this is the first time you've heard the word rhizophagy. I want to know how many of you are discovering this today. Dr. White's team didn't stumble onto rhizophagy by accident. They were investigating something that had puzzled plant scientists for years: why plants grown in sterile mineral-complete nutrient solutions often underperformed compared to plants grown in living soil, even when every measurable nutrient was present in the sterile solution. The numbers were there. The growth wasn't. When they began examining root cells under confocal microscopy, a technique that uses laser light to image individual cells in 3 dimensions, they found bacteria inside the root cells themselves, not on the surface, not in the spaces between cells. Inside the cells, living there, being processed there. This was not contamination. This was not infection in the pathological sense. This was a structured, repeatable biological relationship. What followed was years of careful experimentation. They inoculated sterile-grown seedlings with specific bacterial strains and watched what happened. Grass seedlings inoculated with endophytic bacteria, bacteria capable of living inside plant tissue, showed measurably better growth than uninoculated controls, even when both groups had identical mineral nutrition. The bacteria weren't just passengers, they were contributing something the mineral solution alone could not provide. Further work identified the reactive oxygen species mechanism, the chemical stripping of bacterial cell walls, as the key to how bacteria enter root cells. In the first place, this was a significant finding because reactive oxygen species are typically associated with plant immune responses. The plant's own defense chemistry, it turns out, is also its harvesting tool. The same molecular machinery that fights pathogens is being repurposed to process beneficial microbes for nutrition. That dual function is elegant in a way that should make every synthetic fertilizer manufacturer deeply uncomfortable. Because what it suggests is this: a plant's nutritional capacity isn't fixed by the chemistry of its soil. It's dynamic. It scales with the diversity and density of its microbial community. Enrich that community, and the plant's ability to feed itself expands. Destroy it, and no amount of dissolved minerals fully compensates. This is the core argument of rhizophagy research, and it's one that the broader agricultural industry has been very slow to absorb. per poor wood, or your broader Rhizophagy and press. Rhizophagy doesn't operate in isolation. To understand it fully, you have to understand its relationship with the mycorrhizal network, the fungal web that connects plant roots across distances that individual roots could never reach alone. Most gardeners have heard of mycorrhizae by now. The fungal threads, hyphae, extend outward from root surfaces and dramatically increase the effective surface area available for nutrient uptake. A single teaspoon of healthy forest soil can contain several kilometers of fungal hyphae. That's not a metaphor, that's a measurement. What the mycorrhizal network does, in part, is deliver bacteria. The fungal hyphae act as highways, transporting microbial passengers from distant soil zones back toward the root tips, where the rhizophagy cycle begins. Research has documented bacteria traveling along high-fall surfaces, essentially hitching rides toward the plant. In exchange, the plant feeds the fungi with carbon, sugars produced through photosynthesis, pumped down to the roots and out into the fungal network. This is a three-way economy: plant, fungi, bacteria, each providing something the others need, each dependent on the others to function at full capacity. Here's where it gets practically important for anyone growing food. Mycorrhizal associations form with the vast majority of land plant species. Estimates suggest somewhere between 80 and 90% of all plant families engage in these relationships, but there are critical exceptions. The Brassica family, your cabbages, kale, broccoli, mustards, does not form mycorrhizal associations. Neither do most plants in the Chenopodiaceae family, which includes spinach and beets. This doesn't mean rhizophagy doesn't occur in these plants. The evidence suggests it does, independently of mycorrhizae. But it does mean that the soil management strategies you use for your brassicas need to account for the absence of that fungal delivery network. Direct bacterial inoculation of the seed or root zone becomes more important when the fungal highway isn't available. Experienced growers know this intuitively even when they don't have the vocabulary for it. Brassicas respond dramatically to compost incorporation and bacterial inoculants in ways that can't be explained by mineral content alone. Now, you know why. This is the section that's going to make some people uncomfortable. And I want to be precise here because the easiest thing in the world is to make vague claims about chemicals bad, nature good. That's not what this is. The disruption of rhizophagy by modern agricultural practices isn't a conspiracy. It's a consequence. A consequence of optimizing for one variable, immediate yield, while measuring nothing about the biological infrastructure that makes long-term fertility possible. Let's go through the specific mechanisms. High-dose synthetic nitrogen applied as ammonium nitrate or urea suppresses the rhizophagy cycle through a feedback mechanism. When nitrogen is abundant in the soil solution, the plant reduces its investment in the microbial recruitment process. Why send out a bacterial workforce when the mineral is already dissolved and available at the root's surface? The plant is not being foolish, it's responding rationally to immediate conditions, but the consequence is that the microbial community, no longer being cycled through the root system, begins to decline. Diversity drops, population density drops. And when the synthetic input is removed or reduced by economics, by policy, by the farmer's choice, the biological infrastructure to replace it is no longer there. This is the dependency cycle that makes conventional farming so difficult to exit. Fungicides present a different problem. Broad-spectrum fungicide applications used routinely in conventional grain production to protect against foliar diseases inevitably reach the soil and suppress mycorrhizal fungi. Since mycorrhizal hyphae are the primary transport network for bacteria reaching the root zone, fungicide-driven mycorrhizal suppression indirectly reduces the inputs available for rhizophagy. The plant's bacterial supply chain gets cut. Tillage is the third major disruptor. Mycorrhizal networks are physical structures. They take time to build, weeks to months to establish functional connections between plants. Frequent deep tillage physically destroys those networks, forcing the system to rebuild from scratch with every growing cycle. The soil biology never reaches the complexity needed to support robust rhizophagy cycling. None of this means conventional farmers are doing something evil. It means they were handed a set of tools and told to optimize for bushels per acre, and nobody measured what was happening underground. That measurement gap is what researchers like Dr. White are now trying to close. If what we've covered so far, the bacterial harvesting, the mycorrhizal delivery network, the way modern inputs quietly dismantle the whole system, if that's already changing how you think about your soil, then hit the like button right now. Not because I'm asking for a favor, because this kind of deep soil science is exactly what this channel exists to bring you, and the algorithm needs to know you want more of it, because what's coming next is the part that actually changes what you do in your garden. We've covered the science, now we're going to cover the practice, what you can do right now with the soil you have to activate and protect this cycle in your own growing space. Subscribe if you haven't yet, because the next few sections are where the theory becomes actionable, and that's where most videos stop, and where we're just getting started. Let's talk about what you can actually control. The rhizophagy cycle depends on three things being present in your soil: a diverse bacterial community, a functional mycorrhizal network, and a plant root system that is not being biochemically short-circuited by excess available nutrients. Disrupt any one of those three legs and the whole system weakens. The most common way home gardeners disrupt it is also the most well-intentioned: over-fertilizing. When you apply high nitrogen fertilizers to your vegetable beds, especially water-soluble synthetic forms, you're essentially telling your plants to stop recruiting. The bacterial workforce gets laid off, the mycorrhizal network, no longer being fed carbon by a plant that's busy absorbing dissolved nitrogen, begins to thin. And because these networks take weeks to rebuild, a single heavy synthetic application can set back your soil biology by an entire growing season. This doesn't mean never fertilize. It means understand what you're trading when you do. The practical alternative that experienced organic growers have used for generations, and that soil biology research now supports mechanistically, is compost, not as a nutrient source in the conventional sense, but as a microbial inoculant. Good finished compost contains billions of bacterial cells per gram, including many of the endophytic strains involved in rhizophagy cycling. When you incorporate compost into your beds, you're not just adding organic matter, you're restocking the workforce. Biochar is another tool worth understanding in this context. Charred organic material, produced through pyrolysis, the burning of biomass in low oxygen conditions, creates a porous carbon matrix that serves as habitat for soil bacteria. The pores in biochar are sized at the same scale as bacterial cells, providing physical protection from predation and desiccation. When biochar is charged with compost before application, a process called activating, it introduces both the habitat and the inhabitants simultaneously. The key practical principle here is this: feed the biology, and let the biology feed the plant. Every input decision you make in the garden is either supporting or suppressing the underground economy that rhizophagy depends on. Once you see it that way, the decision framework changes completely. What's the one thing you're doing in your garden right now that you're rethinking after hearing this? Tell me in the comments. I read every single one, and I want to know what's shifting for you. Walk into any garden center or browse any agricultural supply catalog and you'll find bacterial inoculants, products containing Bacillus subtilis, Pseudomonas fluorescens, Azospirillum brasilense, Rhizobium species. The labels make impressive claims. The science behind some of them is real, but the application of that science in commercial products is wildly inconsistent, and understanding the difference could save you significant money. Here's what the research actually shows. Rhizobium inoculants for legumes, the bacteria that form nitrogen-fixing nodules on bean, pea, and clover roots, have a genuinely strong evidence base. The mechanism is well understood. The practical benefits are measurable and consistent. If you're growing legumes and your soil doesn't have a resident population of the appropriate rhizobium strain, which is common in soils that haven't grown that legume family recently, inoculation at planting can meaningfully improve yields and nitrogen fixation. This is not marketing. This is documented agricultural science going back over a century. Mycorrhizal inoculants are more complicated. The products are real, the fungi are real, and the science of mycorrhizal symbiosis is solid. But here's the problem. If your soil already contains a healthy native mycorrhizal population, which any soil that has grown plants and hasn't been sterilized likely does, adding commercial inoculants may provide limited additional benefit. The native strains, adapted to your specific soil chemistry and plant community, will often outcompete introduced commercial strains. The practical implication: mycorrhizal inoculants are most valuable in genuinely depleted soils, post-construction sites, heavily fumigated agricultural ground, or container media that started sterile. In a well-managed garden with regular compost additions and minimal fungicide use, the money spent on mycorrhizal inoculants might be better spent on more compost. General bacterial inoculants, broad-spectrum products containing multiple Bacillus or Pseudomonas strains, occupy a murkier space. Some of these strains have documented plant growth-promoting properties, but the shelf life of live bacterial products is highly variable. The viability of cells at point of use is rarely tested by consumers, and the specific strains that participate in rhizophagy cycling are not always what's in the bottle. The most reliable inoculant source: healthy living soil from a garden that's been managed biologically for years. A handful of that soil introduced to a new bed or container carries a complexity of microbial life that no commercial product currently replicates. Experienced growers have known this for generations. The science of rhizophagy now explains why. Here's the part of the story that should genuinely humble anyone who thinks modern agriculture invented the idea of soil management. Long before Dr. White's confocal microscope images, long before the term rhizophagy existed in any scientific literature, farmers around the world were managing their soils in ways that we now understand through the lens of this research. We're actively protecting and enhancing the biological cycles we've been describing. They didn't have the vocabulary, they had the results. Korean natural farming, a system developed and codified by Cho Han-kyu in the mid-20th century but drawing on centuries of East Asian agricultural tradition, centers on the collection and amplification of what practitioners call indigenous microorganisms, IMO. The practice involves collecting forest soil from the base of old trees, where microbial diversity is highest, fermenting it with rice and brown sugar to amplify the population, and then applying it to growing beds and composts. From a rhizophagy perspective, this is precisely the right intervention. Forest floor soil contains exactly the kinds of bacterial diversity, including endophytic strains, that the rhizophagy cycle draws on. The fermentation process selects for facultative anaerobes and beneficial fermentative bacteria while suppressing pathogens. The result is a high-density, diverse microbial inoculant that costs almost nothing to produce and outperforms most commercial alternatives. Traditional Japanese rice cultivation systems that integrated ducks and fish into flooded paddies were, among other things, maintaining bacterial populations in the water column through the constant disturbance of organic material. The duck and fish waste fed bacterial growth. The bacterial growth fed the rice through exactly the mechanisms we now understand. The farmers knew the system worked. They measured it in yield and in the health of the water. They didn't need a peer-reviewed paper to tell them the mechanism. The Amish and traditional Mennonite farming communities in North America have maintained practices, deep composting, cover cropping, minimal tillage, livestock integration that preserve soil biology across generations. Farmers who have worked those soils for decades will tell you that the ground works differently than conventionally managed land. It holds moisture better. It recovers from drought faster. Crops establish more vigorously without the same level of external inputs. What they're describing, in practical terms, is a soil with an intact rhizophagy cycle. This is not mysticism. This is biology that traditional agriculture preserved through observation and practice, and that modern science is now catching up to explain. We are standing at an unusual moment in agricultural history. The scientific framework to understand what healthy soil actually does, not just chemically, but biologically, is finally catching up to what observation-based farming traditions preserved across centuries. And the implications run in two directions simultaneously, toward large-scale agricultural policy and toward what you do in your garden this weekend. Let's start with the big picture. The global food system currently depends on synthetic nitrogen produced through the Haber-Bosch process, a method that consumes roughly 1 to 2% of the world's total energy supply annually. That nitrogen applied to fields at scale is doing exactly what we described earlier, suppressing the plant's own biological nitrogen acquisition mechanisms while simultaneously degrading the soil infrastructure that makes those mechanisms possible. We are spending enormous energy to deliver a nutrient that, in a biologically intact system, the plant could largely acquire for itself. Rhizophagy research opens a credible pathway toward reducing that dependency, not eliminating synthetic inputs overnight. That's not realistic at current agricultural scale, but progressively rebuilding soil biology to the point where external inputs can be reduced without yield collapse. Several research groups are now working on this transition pathway, including work being done through the Rodale Institute's long-running farming systems trial, which has been comparing organic and conventional systems since 1981 and documenting the multi-decade trajectory of soil biology under each management approach. For you, the grower with a garden or a small farm, the actionable takeaway is actually simpler than the science suggests. Stop sterilizing every time you reach for a fungicide, a broad-spectrum insecticide that reaches the soil, or a high-solubility synthetic fertilizer. Ask what it costs the biology underneath. Not every application is wrong. There are real pest and disease situations that require intervention. But the default assumption that inputs are neutral, that soil is just a mineral medium, that biology is irrelevant to plant nutrition, that assumption is demonstrably false. Start feeding the cycle. Compost, cover crops, minimal tillage, diverse plantings that support diverse microbial communities. These are not new recommendations. What's new is that we now have a mechanistic explanation for why they work that goes far deeper than organic matter is good. The organic matter is feeding a biological cycle that your plants are actively running. You're not just improving soil structure, you're restocking a workforce. And here's the thing that I want to leave you with, because it changes the emotional relationship with the act of gardening. When you plant a seed into living soil, into ground that has been managed to support biological diversity, you are not placing an organism into a passive mineral substrate and waiting for chemistry to happen. You are introducing that plant into an active community. A community it will immediately begin to interact with, recruit from, and depend on. The plant will send signals, the bacteria will respond, the fungi will carry messages between them. The whole underground system will reorganize, at least partly, around the needs of that new plant. That's not poetic exaggeration. That's what the microscopy shows. And if you've spent years fighting your soil, adding more inputs, chasing deficiencies, wondering why your plants never quite look the way they should despite everything you're doing, the answer may not be in what you're adding. It may be in what you've been inadvertently removing. The biology was always there. It's waiting to come back. If you've made it this far, through the cellular mechanics, the mycorrhizal networks, the historical systems, the practical implications, you're clearly not someone who's satisfied with surface-level content. That's exactly who this channel exists for. Every week we go this deep into the science of how plants, soil, and agricultural systems actually work. Not the simplified version, the real version, with the mechanisms, the history, and the honest acknowledgement of what we don't yet know. Subscribe if that's the kind of content you want more of, and tell me in the comments what's the one thing from this video that you're going to change in how you manage your soil. I read every comment, and the most common answers shape what we cover next. Your question might be the next video. The next time you push a seed into the ground, you'll know exactly what's waiting for it down there, and why that invisible workforce matters more than anything you could buy in a bag. Thanks for watching. The end screen has more videos in the same world. Pick whichever video on the end screen calls to you. There's more like this, waiting.